Thank you for coming to the Affymetrix workshop. I'm going to spend 15 minutes. I know we want to get to the speakers. I'm going to spend about 15 minutes talking about new capabilities in our expression and genotyping portfolio. On our expression side, we have introduced updates as well as new piece of software within expression console software. So there are some new features that I'm going to talk about, and then there's a new software which is the Affymetrix transcriptome analysis console, following along the same lines as genotyping console and the other software that Affymetrix provides on its website. The improvements in the expression console software include principal component analysis now. It will help identify outliers using the PCA algorithms, checkpoints in PCA graph to highlight samples, and then samples can be labeled by color, shape, and user attributes, which are available from your sample ARR file. We've also added new features to show the image data as thumbnails, and so the key here is that you can see multiple, and with a single click, you can open and do a quick QC analysis of the cell files that are coming out of the system. From the expression console, you can also launch the transcriptome analysis console software. And the key here is we want to make things as easy as possible with as few clicks. And the transcriptome analysis software itself, we've tried to make that very easy. There are basically 4 steps for differential expression. And the goal here is that we want to get to the answers in as few steps and make it extremely easy to use. So you don't need an expert bioinformatician to perform all of the analysis. So there are 4 steps here. You start with the first step, which is you specify the analysis that you want to perform. Your second step is you import your chip files. It includes the attributes are automatically included so that you can select the condition step. You specify the condition names, or you can simply drag and drop the files over here into a condition group. And then finally, you run the analysis. So from input to answers in 4 easy steps, which gives you then a view with the genes that have been differentially expressed. So over on the left side, you see the genes that are upregulated and downregulated. In this case, it was an experiment where we had control versus the case where it was dye versus wet growth. You can see which genes are upregulated, which genes are downregulated. On the right side, it shows you different plots. So you can see our cluster plots over here. You can use lasso tools to identify the differentially expressed genes. And then you can establish different algorithm options or filter criteria. Really, what we're trying to do over here is try and get to your answers as quickly as possible. Once you have selected your genes, you can sort and export those differentially expressed genes into Excel or any other software. The software itself is available on our web. If anyone has questions, please visit our booth. There will be people there to show the software as well as answer any questions that you may have on this. From a model organisms perspective, we continue to introduce new arrays. We'll have a tomato, a rabbit, a potato, guinea pig, and then we're working with the International Wheat Genome Consortium to come up with a wheat expression array as well. If anyone is interested in any of these model organisms, especially the wheat array, we have Stan Trask over here. He works in the expression strategic marketing, and he can help answer questions. He can also gather any input that you may have on these arrays. We also have a number of community arrays that are already available. These are available on the website. There is a pamphlet available on our booth, and on which you can see it's maybe a little difficult to read it from the back over there. The new community arrays that are being planned also, we have a plasmodium, a lettuce, and pepper, and then on the expression side, we have sorghum, tobacco, switchgrass, and rhodobacter. With this, I want to go to the next step, which is genotyping solutions, and we made numerous improvements within our genotyping portfolio. I want to give a brief idea about the improvements that we have made in our Axiom Genotyping Solution. Talk a little bit about the software and the bioinformatics that's available with the Axiom Genotyping Solution. So the Axiom Genotyping Solution is a complete solution that is a complete end-to-end, offers complete end-to-end workflow for every part of your genotyping program. So if you have content from SNP discovery, that's done through your NGS efforts, or whether you are interested in content that we have in our Axiom genomic database, you can select that and then put it on arrays. These are 96 arrays on a single microtite plate. So you can interrogate 96 samples at the same time in a single process run. The arrays themselves can be customized, or they are pre-designed. And then we offer a complete genotyping solution with a robust assay and reliable assay, the Axiom 2.0 assay. Our target preparation methods are manual or automated protocols, and then this is then processed within the Genotyping MC instrument for hands-free automation. So once you've completed your target prep, you load these array plates with your samples into the system, and 2 days later you have your results that can be analyzed with Genotyping Console software. I want to talk a little bit about the manufacturing process that we use and the advantages of going with our custom assays. The advantages, we use semiconductor-based photolithographic technology. What that means is we use a silicon-based wafer over here, for example. This is in our manufacturing facility right here in Singapore, actually, where they're inspecting the wafer. From this, we actually create these individual arrays. Because we use photolithographic technology, we can actually access every marker that has been designed on the array. So there are no SNP dropouts. A second advantage is we use, because of the photolithographic technology and the Axiom 2.0 assay, you get more marker selection flexibility. What does that mean? So if you have a candidate SNP, then it allows our Axiom design process basically allows you to design for this candidate SNP even if there is a neighboring mutation within 10 base pair from the candidate SNP. In addition, we have proven indel capabilities, and then you can have multi-species design on a single array. So if you're interested in, let's say, rice and wheat on the same array, or if you're interested in 3 different species, then like maybe pig and bovine, then you can design a multi-species array on this Axiom platform. We've put in a lot of effort into improving the genotyping analysis, and there's a reason for that. We want to make the process as easy so that whether it's a researcher or whether it's a breeder, we want the answers to be available as soon as the data is available from the system. We want to make the answers easy to understand, easy to interpret. So we have put in a lot of effort into automated analysis of both diploid and polyploid organisms, something that's not possible today on other technologies. There's absolutely no manual editing required when the analysis is performed, and that gives you the advantage of a hands-free workflow where you can go from your sample to answers in as few as 4 days. I want to talk a little bit about the Axiom assay, which I mentioned. It's a highly specific 2-color enzymatic assay, and the way it works is we have oligonucleotide probes. These are 30-mer probes that are designed on the array. They're very specific to the genomic locus, and then they are photolithographically synthesized on the microarray. With that, we take the sample. We start with between 100 to 200 nanograms of your input DNA. This is then whole genome amplified, fragmented, and stringently hybridized onto the microarray. So we use a ligation-based assay. So what that means is the polymorphic base is interrogated by random 9-mer probes, which are then the DNA ligase then specifically ligates these reporter probes on the array surface. And then the multi-layer signal amplification performs detects the polymorphism that is being interrogated. So because of the stringent conditions that we use for the hybridization, the wash conditions that we use, as well as the design capabilities within Axiom 2.0, we are able to apply this technology specifically for plant and animal genomes, where you experience genomes that are diploid, polyploid, have repeat elements, various complexities that are challenging for other technologies. From the software side, so this is an easy workflow slide, which basically shows once you go through your target preparation and your data is available, you have two options. You can use either the Genotyping Console software, which is a Windows-based software, and you get your results. Or you can use a command line-based software called Affymetrix Power Tools, which can be used on multiple operating systems. We have Windows, Linux, both 32 and 64-bit, and then you can get your results. And then finally, you have an R-based package called SNP Polisher. Once you have your results from, let's say, a 50,000 SNP array or a 100,000 SNP array, what we want to do is present those results in an easy way for the end user, whether it's the breeder or whether you're performing research. You want to look at it in a simple way and look at what SNPs are in which category. So this SNP Polisher software, which I'll talk about a little more in the next slide, categorizes the results that have been discovered through either the algorithms that have been run in Affymetrix Power Tools or Genotyping Console. Both software use the same algorithm, so it's your choice. If you're interested in a high throughput mode, then you can use Affymetrix Power Tools. With the Windows-based software, you can analyze data plate at a time. Again, the goal here is, much like I mentioned in our expression console and the transcriptome analysis console software, you want to make this as easy. So go to your results in as few as 4 steps again. And that's the key over here. So you group your samples into batches and perform sample QC. You genotype your selected samples, and then you can QC the SNP and genotype and classify them into 6 types. And then additionally, you can provide what's known as off-target variant genotyping, which is available in the SNP Polisher. About that. The key here is there's no manual editing required. They're tunable parameters, so whether you have polyploid or diploid, inbred or non-inbred samples, you can control that. If you have if you're working with a polyploid genome, then you have polyploid-specific priors, which basically allow you to go to your answers without any manual editing. There were 3 abstracts that we presented at the Plant and Animal Genome Meeting in North America. You can download these abstracts from our website when you go to the Agri Genotyping landing page. Feel free to stop by the booth or meet with one of us, and we'd be glad to go through that. The SNP Polisher software, as I mentioned, so once you have your results, how do you look at it? You have, probably working with 3,000 samples, maybe it's 1,000 samples, it's a 50,000 array. Well, the SNP Polisher software breaks down that analysis. It breaks it into 6 simple categories. First category is poly-high resolution, and what that means is good cluster resolution, and there are at least 2 examples of the minor alleles. So you have 3 clusters here, nicely separated. No minor homozygous is basically 2 clusters with no examples of the minor homozygous genotypes. And you typically see this if you have, let's say, genotype only about 48 samples. As you increase your sample size, you start the SNPs from the no minor homozygous will start showing up in the poly-high resolution. Then you have the mono-high resolution, where you see only 1 cluster, and that's typically less than 2 examples of the minor allele. We've introduced what's known as off-target variant genotyping within the SNP Polisher software. And in here, what you see is actually a 4th cluster. And typically, normally, any ordinary genotyping console software, genotyping analysis software would have classified it or would not have known what to do with this 4th cluster. This 4th cluster is typically what you would have called null alleles in microsatellites. It's caused by double deletions or sequence non-homology, where your sample differs from the reference genome. And SNP Polisher software now allows you to pull these samples out separately, so you can analyze the 3 clusters, and then there is this 4th cluster. And then call rate below threshold is somewhat important in plants because when we do our analysis, we set a call rate threshold of 97%. And then we start classifying the clusters. Sometimes these clusters are well resolved, but they fall below our call rate. And typically in plants, you might want to set a threshold of maybe 95% because those clusters are still, because those SNPs are still important. Well, call rate below threshold is that category. And you can tune those parameters, and you can say, well, I want to have everything about 97 fall into this, or I want everything about 95 to fall into, everything under 95 to fall into this category. So it makes it very easy to break it up into the 6 categories. And then finally, the last category is the other category, where typically we show clusters that are not well resolved even by the eye. What makes the analysis so special in genotyping analysis? Over here we have, this slide basically shows our capabilities for polyploid analysis, and you have polyploid SNPs. And this is just a model which shows where is it shows basically what can be expected when you have 2x, 4x, 6x, or 8x. And so you have simulated versus observed patterns. In this case, this is a simulated pattern. And in a normal diploid species, the river buffalo I've shown over here, you have 3 nice clusters separated out. But when you go into something like an allotetraploid, which is 4x, this one is from rapeseed, you can actually see 3 clusters, but there's a fair bit of compression, and these clusters have moved to the left. And that can throw off other genotyping software from performing the analysis. And that's when you need to go in and do it manually. Over here, within our genotyping console software or the APD tools, all of this is done automatically. So you don't have to go in and edit anything. This example is from wheat. Again, you see the same kind of, the clusters are shifted to the right, and they're slightly compressed, I would say. More compressed than the rapeseed. So why does it make it, why is it so important to have automated? And I'll give you an example over here. So assume you have, this is what your results come out of the algorithm. This is without any clustering, without any genotypes being assigned. There are plenty of clustering, automated clustering algorithms. If the eye can resolve this very nicely, you see these nice 3 clusters, and you can actually assign the genotypes quite easily. But what happens if, let's say, the clusters are shifted? Will your software be able to assign the genotypes correctly? Will you have to go in and manually edit the genotypes? All these things become important. And when you are working with maybe 600 SNPs, it's not such a difficult task. But when you start working with 1,000 SNPs, 10,000 SNPs, you can think how challenging this would be if you had to go in and manually assign a genotype or correct the problem. And that's where we put a lot of emphasis in our software, where the Affymetrix genotyping software not only automatically clusters it, but can automatically assign the genotypes as well. And the key here is your technology able to detect the heads in here? Is it, unlike other platforms which might have missing data, which might have SNP dropouts, the Axiom technology is extremely robust, allowing you to pick out the heads from the plant samples. Something very important when you're working with plant genomes. And then over here I have other representative cluster plots. This is from a wheat array that was developed in collaboration with some of our European colleagues. How does it work? We have worked with Dr. Woods at the Wageningen University and modified his algorithm, which is called FitTetra, which to make genotype clustering in an hexaploid situation. What we do is basically we start out with 12 initial priors in both what's known as the contrast and the size dimension, and then we obtain posterior locations for each cluster. And this is a typical workflow for a polyploid genome. And then the Axiom GT1, which is the algorithm that's in our genotyping console, can proceed with the genotyping with the posterior cluster location. So to give you an example, this is what happens. So once you have your data, we know whether it's a diploid or polyploid based on the analysis parameters that we provide. If it's a diploid, it's it's a simple 3-pattern cluster, and your algorithm, the Axiom GP1 algorithm can detect those 3 clusters, as I showed previously. If it's a polyploid, then we have the fit algorithm, which is modified to produce these genetic SNP-specific prior. And then these are used by our Axiom GP1 algorithm to come up with the right clustering algorithm. All of this is done automatically. Absolutely no manual editing is required. So it allows you to analyze thousands of SNPs in about an hour. This is an example pilot project that we ran in late 2011. This was work that we did with Indra. There was a similar pilot project that we did with the University of Bristol. In which case, what we did with this pilot project, we had about 18,000 SNPs. Some of them were validated with Caspar. We designed an array, and then we did a wet lab experiment with within Affymetrix in our services lab. And then we genotyped about 408 lines, and these were about 238 elite varieties plus 170 mapping populations. The results are shown over here. So there's about 77% that were automatically genotyped, and they fell into these 6 categories, with about 39% in poly high resolution, 2.7 in the no minor homozygous, about 38% in mono high resolution, and then about 1% in the off-target variants. And that was the key over here with the lead data, being able to automatically genotype a complex polyploid. Genotyping of allopolyploids is very different than autopolyploids. Allopolyploids, such as wheat, we look at, there are typically 3 clusters, where we are looking at the SNP that is fixed in one genome, or is segregating in one genome relative to the other 2 genomes. In autopolyploids, you actually see 5 clusters, and this is the simulated model, again, that I had shown previously. And we can actually what we're working on now is the ability to automate more than 3 clusters using the fit tetra algorithm that I spoke about a little earlier. So this example is from a rows where you see allele dosage ratios. So you can see the different genomes contributing to the cluster over here. And there are 5 well-resolved clusters which the automated genotyping algorithm has been able to detect. Some additional plots from the rows array, where you can say 5 different clusters, and this is where it becomes important. You're working with thousands of SNPs, absolutely no user intervention. At the end of the results, you're presented with data that has been automatically clustered and genotyped. So Affymetrix SNP genotyping technology, we've evolved quite a bit. We started our collaboration with Dr. Susan McCutch on the RICE44K. We've learned quite a bit from there. We've designed multiple species on our cartridge arrays. And then when we launched Axiom Genotyping Solution in 2009, we've designed a number of custom arrays. This includes bovine, chicken. There's a paper that was released just earlier this month. A copy of it is available at our booth if you're interested. It's an extremely nice paper. It talks about NGS discovery, how it was applied, and how the SNPs were screened and then designed into a final array. We also designed a custom sunflower. Dr. Williams here is going to talk about the buffalo, which we designed in collaboration with the International Buffalo Genome Consortium. We also designed a custom catfish and carp array. There's a poster on the carp array actually right outside this room, poster 93, so if you're interested you can take a look. It talks about how our arrays are designed. I already mentioned the rows. There's a breed array that we've designed. What I showed you was the pilot experiment. Subsequent to that, we've designed a custom 420,000 SNP array for the Breed Wheat Consortium. This is a private consortium out of Europe that requested the design from us. There's a strawberry array, and then in the chrysanthemum array, these are all custom arrays. The chrysanthemum is custom, and in the future, we'll have an equi and a salmon, and then a maize array. So plenty of arrays from Affymetrix, and the key here is they all, the reason the customers have gone with us is primarily because of the analysis. There's no missing data, no SNP dropouts. You can detect your heads quite easily. The analysis is extremely easy, and that's the paradigm of Affymetrix, that we want to make the bioinformatics extremely easy, whether it's expression or whether it's genotyping. And last, I want to talk about a new product that we're launching in a couple of weeks. This is 384 arrays. I showed you 96. So the 384 arrays, basically, there's a single array over here, allows you to interrogate 384 samples at the same time. You can have up to 50,000 SNPs for diploid species, 32,000 for polyploid, and you can go 3,000 samples per week for a throughput. So now you have the ability to use the Axiom Genotyping Solution for every phase of your study, whether it's discovery, trait association, or routine test. You can go all the way from 2.6 million markers per sample down to 50K or 1,500 markers with up to 384 samples per week. With that, I'd like to thank you for listening to the new capabilities from Affymetrix.